name is Jesse, and this is my plasma membrane video. The plasma membrane, a protein-studded fatty film so thin that it cannot be seen directly in the light of a microscope. It is made up of a phospholipid bilayer, which is the area in the middle of the membrane composed of a phospholipid head and a hydrophobic tail. The hydrophobic tail consists of either an unsaturated fatty acid chain or a saturated fatty acid chain, which is a saturated fatty acid is a fatty acid such as a stearic acid whose carbon chain contains no unsaturated linkages between carbon atoms and hence cannot incorporate any more hydrogen atoms. An unsaturated fatty acid is a fat or fatty acid in which there is at least one double bond within the fatty acid chain. The plasma membrane has membrane channels, which is a family of biological membrane proteins which allows the passive movement of ion channels, water, aquaporins, or other solutes to passively pass through the membrane down their electrochemical gradient. I put simple diffusion there because simple diffusion is a process whereby a substance passes through a membrane without the aid of an intermediary such as an integral membrane protein. This leads to selective permeability because it picks and chooses basically what goes through. That leads me to vesicles. Vesicles are a mechanism of transport, which is attached to passive transport. Passive transport is the spontaneous movement of a solute down its concentration gradient across a cell membrane via a membrane transport protein such as a channel or a transporter, which I attach to the channel-mediated passive transport, which is the process of spontaneous passive transport of molecules or ions across a biological membrane via specific transmembrane integral proteins. And then I put all the transports basically together, which leads to active transport. And the active transport is the movement of a solute across a membrane against its electrochemical gradient. From active transport, I put osmosis, which is passive movement of water across a cell membrane from a region where the concentration of water is high to a region where the concentration of water is low. This led me to the solutions. You have isotonic, hypertonic, and hypotonic. From vesicles, I went into ion channel and gated ion channel because the vesicles move through those. The ion channel is a transmembrane protein that forms a pore across the lipid bilayer through which specific inorganic ions can diffuse down their electrochemical gradient. And the gated ion channel is a transmembrane protein that forms a pore across the lipid bilayer through which specific inorganic ions can diffuse down their electrochemical gradient. That led me to the gradients, which was the electrochemical gradient and the concentration gradient. The electrochemical gradient is the driving force that determines which way an ion will move across a membrane. The concentration gradient is the process of particles, which are sometimes called solutes, moving through a solution or gas from an area with a higher number of particles to an area with a lower number of particles. As I go back to the top, plasma membrane, I have a branch off of the membrane proteins. The membrane proteins are Membrane proteins are a protein associated with the lipid bilayer of the cell membrane. This led me to list off all the proteins that were given. Integral membrane protein, peripheral membrane protein, enzyme membrane proteins, receptor membrane proteins, anchor membrane proteins, and transporter membrane proteins. Integral membrane proteins are a type of membrane protein that is permanently attached to the biological membrane. Peripheral membrane proteins are membrane proteins that adhere only temporarily to the biological membrane with which they are associated. Enzyme membrane proteins are a type of protein that is attached to or associated with a biological membrane. Receptor membrane proteins are receptors that are embedded in the membrane of cells. Anchor membrane proteins are linked intracellular actin filaments to extracellular matrix proteins. And transporter membrane proteins are proteins that move the solute across a cell membrane by undergoing a series of conformation changes. From those membrane proteins, I attached glycoprotein and proteoglycan. Glycoprotein, any protein with one or more covalently linked oligosaccharide chains. And proteoglycan is molecule consisting of one or more chains attached to a core protein. From the membrane proteins, I attached the protein-attached membrane-associated proteins, the lipid-linked membrane-associated proteins, and the monolayer-associated membrane proteins. The protein-attached membrane-associated proteins is proteins that interact with or are part of biological membranes. They include integral membrane proteins that are permanently anchored or are part of the membrane, and peripheral membrane proteins that are only temporarily attached to the lipid bilayer or to other integral proteins. Lipid-linked membrane-associated proteins are proteins located on the surface of cell membrane that are covalently attached to lipids embedded within the cell membrane. Monolayer-associated membrane proteins are proteins that, situated within the cell, or the cystole, and interact with in the inner layer of the lipid bilayer. From lipid-linked membrane-associated proteins, I put glycolipid. And glycolipid is a membrane lipid molecule that has a short carbohydrate chain attached to its hydrophilic head. From the membrane proteins, I also attached the molecules, the detergent and the amphipathic. The detergent molecule is soapy substance used to solubilize membrane proteins. And the amphipathic protein is having both hydrophobic and hydrophilic regions, as in a phospholipid or hydrophilic head. And lastly, I have the models, which is the sandwich model and the fluid mosaic model. The sandwich model was Davison and Danieli, the original model of the membrane presented by Davison and Danieli. The fluid mosaic model was from Singer and Nicholson, the second model of membrane developed by Singer and Nicholson.